Bismillahirrahmanirrahim. Talking about the helminths, they have three classes, namely nematodes, trematodes, cestodes. Nematodes are long roundworms. Trematodes, being leaf-like, are called flukes or flatworms, while cestodes are long ribbon-like worms called tapeworms. The parasite to be discussed today is Strongyloides stercoralis. Strongyloides stercoralis is a roundworm that's a nematode and it causes the disease called strongyloidosis. The common name Strongyloides stercoralis is the threadworm. The adult Strongyloides stercoralis live in tunnels in the mucosa of the small intestine. The male and female worms, they live there, take nourishment and protection. The males, they are smaller and they can grow to only about 0.9 millimeters in length, while females, larger ones, they can grow up to 2 to 2.5 millimeters in length. Talking about the life cycle of Strongyloides stercoralis, there are two types of life cycle in Strongyloides, and the much complex life cycle has Strongyloides stercoralis. In one type of cycle being called free living cycle, the male and female worms living in the tunnels in the mucosa of the small intestine, they made the gravid female lays eggs, and the eggs hatch and reproduciform larvae are produced, which migrate to the lumen of this intestine. And once in the lumen, they have to be excreted in the feces. Once outside the body on the soil, the reproduciform larvae can take two pathways to develop further. In one path, the reproduciform larvae, they develop directly into the filariform larvae, which is the infective stage for human beings. The other pathways, these reproduciform larvae, they develop into adult male and female worms, which the gravid female lays eggs, and these embryonated eggs, larvae develop in these embryonated eggs. They hatch and reproduciform larvae are released. These reproduciform larvae, they develop further in due course of time, and they develop into the filariform larvae, which is the infective stage for human beings. And these filariform larvae are capable of penetrating the intact human skin. The second type of life cycle is the parasitic life cycle. And in this part, what happens is that the filariform larvae, after the intact human skin, reaches small intestine by different routes. Historically, it is believed that they enter into blood vessels, reach the circulation, and by circulation they reach the lungs, they penetrate the alveoli, reach the alveoli, and there are then they travel upwards through the bronchi and trachea up over the epiglottis, and it is swallowed into the esophagus. The larvae pass through esophagus, through the stomach, and reach the small intestine, where they tunnels, where they reside, they take nourishment and protection. The other form, these reproduciform larvae, once produced in the small intestine, they can directly develop into filariform larvae. This is called autoinfection. This completes the life cycle of this parasite, which, to summarize once again, the female living in the small intestine lays eggs, they hatch, Rebritiform larvae are produced, which can develop by these two routes to filariform, which are infective to human beings. And they can penetrate the intact human skin. They penetrate the intact human skin. They reach the circulation, pass through the lungs, and at the end, they reach the mucosa of the small intestine. Having known the life cycle of Strongyloides stercoralis, about the disease caused by Strongyloides stercoralis is called strongyloidosis. Most of the people, many of the people infected by this parasite remain asymptomatic. But when symptoms there, there can be dermatitis, in which there is swelling, itching, and mild hemorrhage at the site of penetration of the skin. While penetrating through the alveolar septa, they can produce pneumonia-like symptoms. And when present in the intestine, there may be burning pain, tissue damage, ulcers may be formed, and sepsis occurs there. And when this happens, there may occur chronic diarrhea in which stools may have yellow mucus in them. About the diagnosis of this disease, the diagnosis is difficult, but mainly the stool examination is done for the presence of larvae in the stools. These stools can be cultured on agar plates, and later on it is examined under the microscope for the presence of larvae. And thirdly, serological tests by enzyme-linked immunosorbent assay method can be done to detect the or antibodies related to this parasite. We're talking about the helminths again. The round nematodes being the round worms like this. Talking about nematodes, and they have got leaf-like shape, and they are called flukes. Nematodes are flukes, and those nematodes which pass some of their life cycle in blood, they're called blood flukes, schistosomes. And nematode parasites which are related to the liver, like Clonorchus sinensis and Fissiola hepatica, they are called liver flukes. And those parasites like Paragonius westermani, which are related to the lung, they are called lung flukes. About blood flukes, the genus which we'll study is Schistosoma. And among this genus Schistosoma, there are three species: Schistosoma hematobium, Schistosoma mansoni, and Schistosoma japonicum. About Schistosoma hematobium, parasite is found in Africa and the Middle East. It's a major agent of schistosomiasis. The disease which is caused by schistosoma is called schistosomiasis. The adult worms of Schistosoma hematobium, they have got cylindrical body 1 to 2 centimeter. There are two terminal suckers, a blind digestive tract, and reproductive organs are there. And by virtue of these suckers, it attaches to the surfaces. Male's body forms a groove or gynecophoric channel in which it holds the longer and thinner females like this. This is the male one, and the longer, thinner female is placed in the groove formed on the ventral side of the male's. Schistosoma hematobium lives within the perivesical venous plexus, that is the venous plexus around the urinary bladder. The schistosomes, all the schistosomes, that is all three genus of schistosomes, feed on blood particles through anaerobic glycolysis. About the life cycle of Schistosoma, hematobium, and other schistosomes, there are two parts in the life cycle. As Schistosoma hematobium resides in the vesical plexus, the eggs, female lays eggs, and eggs are passed in the urine. When eggs are passed in the urine, while on the soil and in water, the eggs develop and hatch, and the baby parasites, myristidia, are produced. And these myristidia, they are ready to penetrate the intermediate host snails, which are usually present in the infected water. After penetrating the intermediate host snails, further development of the parasite occurs, and a stage comes which is called sporocysts. Sporocysts, they are developed within the snail, and then the next stage is cercari. The cercari are released into the contaminated water, and it swims freely there, and it is now ready to infect man. It is ready to penetrate the intact skin of human beings in contact with this contaminated water. And this can happen in the rice fields like this, or in some other way when the man comes in contact with contaminated water. After penetrating the intact skin of a human being, the cercari lose tails during penetration and become schistosomuli. These schistosomuli, they reach the circulation, and through blood, they migrate to portal circulation, portal blood in liver. They mature there into adults, and in case of hematobium, they reach the vesical plexus, there is the venous plexus around the urinary blood, where male and female mate, the gravid female lays eggs, and eggs are into the urine. And this is how the life cycle of this parasite, Schistosoma hematobium, is completed. Schistosoma resides in the vesical plexus, while Schistosoma japonicum resides in the mesenteric plexus, that is the venous plexus in mesentery, and similarly, Schistosoma mansoni also resides in the venous plexus. About the disease produced by Schistosoma hematobium, the disease called schistosomiasis. It includes hematuria, scarring in the urinary bladder, calcification in the urinary bladder, and very rarely squamous cell carcinoma can be produced in the urine. And occasionally embolic egg granulomas can be produced in the brain and spinal cord if these baby parasites reach there through circulation. About the diagnosis of this disease, traditionally diagnosis has been made by examination of the urine for the presence of characteristic eggs of Schistosoma hematobium. Thus, characteristic eggs of Schistosoma hematobium are elongated eggs with terminal spine like this. A skin test is also available to diagnose schistosomiasis, and the presence of schistosoma can also be determined by the presence of antigens or antibodies by ELISA method. Schistosome X, the schistosoma hematobium with terminal spine, schistosoma mansoni with lateral spine here, and schistosoma japonicum with a small lateral spine somewhere here. Similarly, this one. Thank you.